Our next speaker is one of our hosts for today, and we appreciate everything you've done, Prakash, to put on this program. And Prakash is going to talk about progress in the development of a blood stage vaccine targeting falciparum AMA1 with RON2.
Thank you, Carol. Let's see. Okay. There you go. All right, perfect. We are nearing the end of the symposium, and we saved the best for the last, right? I'm kidding. Well, we all care about malaria because we want to cure malaria, and it comes down to hopefully generating the next generation of vaccines, such as what Simon presented on. And I'm going to hopefully piggyback on some of the things that he spoke about and talk about another target. Something that many people would be familiar with, the name AMA1, and some people close their eyes, hearing the name AMA1. And if you're among the group like me, I'm like, okay, there are some opportunities to learn something from the biology of the parasite and hopefully improve on its potential as a vaccine candidate. So this is a beautiful electron micrograph, and I think this was from Dr. Aikawa's iconic image. It shows a falciparum myozoite entering a human red blood cell, and you see all those color-coded organelles that store different parasite proteins involved in invasion. And this is another iconic study from 1960s that showed that immune gamma globulin transfer to children with acute malaria can clear parasitemia and clear the clinical symptoms, right? So this, I think in my mind, is being the goal for a blood stage vaccine, is to be able to recapitulate something like that with the vaccine. Right. Simon alluded to immunogenicity, and Lou was asking questions about Rh5 immunogenicity. There are obviously vaccine candidates that have different immunogenicity, natural immunogenicity, right? So on the left, you see an example for Rh5, which seems to have a pretty low immunogenic potential. On the right, you have AMA1, which seems to be a very immunogenic antigen, but it comes with its own advantages, pros and cons. Owing to the lower immunogenicity, you have less antigenic variation, and AMA1 is one of the most polymorphic antigen, right? So how do we overcome some of those challenges? And prior to going to those, answering those questions, I want to present some of these field trial data. The left side panel is the data from Simon's group that did this beautiful study with vaccinating with recombinant AMA1 and doing a blood stage parasite challenge, right? And you essentially see no difference between the blue groups, which are the AMA1 vaccinated, and the red control groups. And similarly, a field trial in Mali conducted by Chris Plow's group also showed that a recombinant AMA1 vaccine conferred little to no protection. There may have been some strain-specific effect, but in follow-up years, that effect was not seen. So we don't think that was probably very real in that instance. But essentially, it says that these AMA1 vaccines in small animal models and even in non-human primates showed good protection, but in humans, it doesn't seem to work. So what might be the reasons? And again, I'll allude to these 3 points that Simon mentioned as well. One is maybe the antibody quality is not sufficiently good in vaccinating in humans. Maybe the antibody quantity is not sufficient, and obviously the polymorphisms. Polymorphisms in this case may not have been the reason for AMA1 vaccine failure, because in Simon's study, they challenged with the vaccine-type parasites and still saw no protection, right? So polymorphisms are a real issue in the field, but not the reason for why an AMA1 vaccine failed in human clinical trials. So I'm going to just switch gears for the next couple of slides and provide you a little bit of molecular parasitology and trying to understand the function of this AMA1 protein. And the AMA1 protein is conserved in all epicomplexan parasites. And in fact, it was shown in Toxoplasma earlier by Jean-François Dubramet's group that AMA1 forms a complex with a bunch of other parasite proteins called the RON proteins, or the RON complex proteins. And when you do an immunoprecipitation of AMA1, you pull down the entire RON complex. So without going into all the details, what we think is going on is that during parasite, the process of merozoite invasion, it secretes its rhabdoid contents. The RON complex proteins are stored in the neck of the rhabdoids. At some point, it is injected and somehow miraculously inserts itself within the plasma membrane of the red blood cells, where a small part of this RON2 protein, and here on I'm going to refer to that as RON2L or RON2 loop peptide, that binds to a hydrophobic pocket in AMA1, and this binding is essential for the parasites to invade. Okay? That's what is shown here. So in this case, falciparum merozoites, which are treated with cytokinesin D that blocks their actin myosin motor, they are able to attach to red blood cells, reorient and form the junction but cannot invade, but they start secreting their rhabdoid contents into the red blood cell. When you use a specific monoclonals such as AMA1, which also this 4G2 monoclonal antibody, which also appears to block the interaction of AMA1 to RON2, they block the parasite invasion, specifically blocking the step in junction formation. So these parasites that are, if you block the interaction of AMA1 and RON2, they don't form the junction and they cannot enter the red blood cells. So this interaction lends itself to some interesting possibilities, right? One, obviously through vaccines, but potentially you could think of small molecule inhibitors of protein-protein interaction that could be a viable approach to target this interaction. And in fact, through a collaboration with GSK, we are trying to screen for some small molecule inhibitors from their TCAMS collection. And we have some sub-micromolar potency compounds that seems to fit the bill. But today I'm going to focus on antibodies. So the first thing that we wondered was trying to address the question of antibody quality. And the reason for that was when we look at the structure of the unbound form of AMA1 shown on the left and the RON2L bound form that is shown on the right, you can see that there are certain structural changes happening in these loops surrounding the binding site of RON2 on AMA1. So our initial idea was, does these conformational changes, would that have an impact on antigenicity of AMA1 or immunogenicity towards AMA1? So these are studies that are published a while ago. It's essentially to show that when we have a preformed complex of AMA1 and RON2, when you vaccinate mice with this Yolii mouse malaria model, 100% of the mice vaccinated with the complex clear the parasites, whereas all the mice that were immunized with AMA1 alone or the RON2 alone succumb to the disease. And this protection is critically dependent on the formation of a functional complex, because if you immunize AMA1 and RON2 to the same mice on different sites, you don't see that protection, right? So this suggests that you need to have a preformed complex to see this observed protection there. And this was also reproduced in a non-human primate malaria model where the complex induced significantly greater protection than AMA1 alone. All right. So what is happening on there? So when we looked at the antibody responses between the animals that were immunized with the complex or AMA1, we found that there was a leftward shift in this neutralization assay, where at the same antibody titer shown on the x-axis, you get greater neutralizing antibodies targeting the parasite. So this is a definition of a change in the quality of the antibodies, right? And what we were able to find was that there were some regions of AMA1 that were being preferentially targeted, or antibodies against specific regions of AMA1 like 1E and 1F that made more antibodies in the complex vaccinated group compared to the AMA1 group. So we think that these are important critical targets of AMA1 targeting neutralizing antibodies. This is not limited to Plasmodium. As I said, the AMA1-RON2 interaction is important in many epicomplexan parasites. Indeed, a recent study from Maurice Lebrun's lab in Toxoplasma showed that when mice were vaccinated with just AMA1 alone, or the Toxoplasma equivalent of AMA1 in complex with the RON2, they saw significantly greater protection of mice against Toxoplasma infection. And interestingly enough, these mice were also had significantly lower cyst burden in their brain compared to the mice that were immunized with only AMA1 alone. So there seems to be something unique about this antigen in its ability to induce a qualitatively better antibody response. Okay. So keeping that in mind, what we wanted to do, or what we attempted to do over the last few years, is to be able to generate a genetic fusion. So you can imagine this vaccine requires mixing of these two proteins together. They spontaneously come together with a pretty high binding affinity of about 20 nanomolar Kd. but it still requires the mixing of two proteins. And we thought, okay, can we design a fusion protein that would avoid the mixing of these two proteins? And so we engineered this protein where only the small part of RON2, which we call RON2L, is sufficient to bind AMA1. And we replaced this loop in AMA1, which is called the D2 loop, which you won't see it here, which is right there. We replaced that loop with the RON2L peptide to create a single genetic fusion. And we showed that this genetic fusion resembles the structure of the AMA1 RON2 complex and mirrors this very faithfully. And so if you want to know more about that, and along the way, we had some curveballs and some interesting observations, but there's a poster by Sean Yannick, a PhD student in the lab. Please check it out. The second thing, so obviously the AMA1 RON2 complex appears to enhance the quality of antibodies targeting AMA1. And the next question that we attempted to address is, can we increase the quantity of these qualitatively superior antibodies? So how do we go about that? So on the left is shown the structure, the ectodomain of AMA1. It's comprised of domains 1 and 2, which come together to form this pan-apple domain. That is the binding site for the RON2. And a domain 3, which is probably more divergent between epicomplex and parasites. And we knew that only domains 1 and 2 were sufficient to bind RON2. And so here we asked the question, can we skip this using the entire AMA1, the full domains 1, 2, and 3, and just use domains 1 and 2 as a way of immunofocusing the antibodies to those critical parts of AMA1 that we want the antibodies to be generated? And that's what is shown here. This is the ELISA data. And shown in blue are the group of animals that received only the AMA1s, domains 1, 2, and 3, or domains D1 and 2, and AMA1 in complex with the RON2L peptide. And what you see is when you vaccinate with the complex, you get slightly lower antibody response against the AMA1. But when you look at the neutralizing activity of these antibodies, both against the full-length protein as well as only the domains 1 and 2, you get far better quality antibodies. And importantly, just immunofocusing the antibodies to domains 1 and 2 seems to generate much greater amounts of these neutralizing antibodies. All right. So the next thing that we want to take is, can we further increase this antibody responses to AMA1? And obviously, we've all heard about the mRNA vaccine approach and its potential for generating these huge antibody titers. And we are not immune to this approach as well. So what we've attempted to do here in the lab is to build an in-house mRNA expression platform. So we scan through a number of literature looking at 5' UTRs, 3' UTRs, and stuff like that. And there were two questions that we asked. Can we increase the immunogenicity using this mRNA approach? But not only that, but would we see differences in immunogenicity if you had different forms of the antigen? Remember, the COVID antigen, the spike protein antigen, is a membrane-anchored antigen. And there has been some anecdotal evidence that probably membrane anchoring may have contributed to its much superior immune responses. And that's something that we wanted to test here. So we made some mRNA in-house. We can transfect hex cells. These cells make good amounts of protein. You can see the protein being expressed in these hex cells, both the secreted form and the transmembrane form. Here is an immunogenicity data in mice. These were mice that were complex with a cationic liposomal formulation that's commercially available. And so we are comparing the recombinant protein in an Adavax adjuvant versus an mRNA vaccine, either in the secretory form or the transmembrane form. And these were mice immunized intramuscularly with the mRNA or the subcutaneous for the recombinant protein 3 times in 2-week intervals, right? So that's a very narrow spacing of immunization. And this is to facilitate getting sera quickly to be able to test these concepts sooner, right? But obviously dosing, antigen concentration and dosing are going to be important for future experiments. What we find very interestingly is that when you membrane anchor the antigen, you get more than 2-fold increase in antibodies to AMA1, right? And you don't see a very big difference after the second boost between the recombinant protein and the mRNA. And I think that's because of the shortened dosing of these immunization schedules. And if you space it properly, and you can see here, after the first boost, the mRNA vaccine definitely makes 2 times or 2.5 times more antibodies than the recombinant protein. So we think this could be a viable approach. And to membrane anchor the AMA1, we've also screened different transmembrane domains and cytoplasmic tails that would efficiently transport the protein to the surface of this eukaryotic cell. So that's something that we are quite excited about. But all these studies with falciparum AMA1 requires that most of these studies are done with these in vitro GIA or neutralization assays that requires purification of the IgG and testing it in an isolated system. Ideally, we would like to be able to test these vaccine candidates in an in vivo model, and we cannot do all these testing in non-human primates because that's the only, Aotus is the only monkey model that will be conducive for falciparum infections. So what we are trying to build right now is to be able to generate a chimeric mouse parasite model where we are replacing the Berghei AMA1 with the falciparum AMA1. And these are very preliminary data. So we just got this data to show that the PF AMA1 is properly localized to the micronemes. You can see that this is an AMA1 where the Berghei AMA1 is also expressed. And so it is you can see it's nice co-localization. It is processed properly to the 83 and the 66 kilodalton form, just like in falciparum. The falciparum AMA1 is processed in Berghei properly. And it appears to be folded correctly because now you can take the falciparum RON2L peptide and they bind nicely and label these micronemal localized AMA1 in these Berghei parasites, right? So we think we got the first set of this this model. Now we've taken these mouse model, these parasites, and replaced the RON2 with the falciparum RON2 to essentially complement the functional complement of falciparum AMA1 and RON2, and hopefully we can use these mouse models to test our vaccines. How am I doing with time? Okay, so the third thing that we want to address is the polymorphisms. And when it comes to AMA1, it's a very big issue. It's a real issue because an eventual vaccine in the field has to be able to overcome these polymorphism issues. Looking at this, I mean, there are over 62 different sites in this AMA1 protein where polymorphisms have been found. Some sites more than others. So when you do a phylogenetic tree analysis of a full-length AMA1 or just the loops, and what I forgot to mention is most of these polymorphisms that you see are in these loops surrounding the RON2 binding pocket. So the RON2 binding hydrophobic pocket is in the middle of this, and all these polymorphisms surround the loops by targeting this RON2 binding site. So how do we go about building a polyvalent or an AMA1 vaccine that covers all polymorphisms? Some people have taken the approach of using a synthetic DICO approach where they've tried to incorporate as many polymorphisms as possible in 3 different AMA1 proteins, or pick AMA1 from diverging parasites, right? Like very different parasites, so as to cover as many polymorphisms as possible. That still does not do the trick. And the challenge there is, I think, when you compare the full-length AMA1, for example, you see this pairing of the CP806, which is a field isolate, which is closely related to the 3D7 lab strain when you look at all the entire full-length AMA1 sequence. But when you look at only the domain 1 loops that surround the RON2 binding pocket, they are quite divergent, right? They're very different. So it depends, what do you target? And so the approach that we took is to be able to generate 6 different variants to cover as many polymorphisms in all these loops without losing their epitope specificity, meaning all these neighboring polymorphisms are kept intact so that the epitope specificity would not be lost between any of these variants. Essentially, you have 6 synthetic AMA1s where the polymorphisms, the clustering of polymorphisms are preserved, are kept intact, right? As an example, I'm showing here this domain 1D loop. It's also called the C1L loop, where there's a clustering of polymorphisms that have been linked to vaccine efficacy. Essentially, what we've done is to take these variants, and you can see these color codes combinations. These are all coming from mix and match from different parasite origins. And we've tried to keep these epitopes, like, for example, this alpha helical. All the mutations that were found on this alpha helical are preserved together, and the ones that are present on this sort of loop structure here are conserved together. Right? So we've been able to generate these 6 proteins, express them in HEC293 cell, SF9 cells, sorry, and confirm that each of these variants retain their ability to bind to RON2. Pretty good. And we've taken all these 7 or 8 different parasite strains, which have their own unique combination of these the mutations in this 1D loop, and you can see how these match across the actual parasites. None of these variants exactly matches, maybe one or two maybe, but most of them differ significantly from any of the parasite AMA1 sequences. And in this case, I'm just showing 1D, and you can imagine there are many more mutations all throughout the AMA1. All right, so here is a quick rundown of these experiments, thanks to Carol Long's lab, Kazutoyo and Abubakar, who are here, who've done these experiments, where now when you take this mixture of these AMA1 proteins, which we call the polyvalent variant 6 mix of AMA1 by itself, or the PV6 in complex with this single conserved RON2L peptide to form a preformed complex, and you immunize rabbits, you see that for every parasite pair, you see that the polyvalent complex mixture always has greater neutralizing activity than the corresponding AMA1 group. And this suggests that when you vaccinate with a polyvalent AMA1 RON complex mixture, you are enhancing the strain transcendency of the antibodies that are generated. And we've been able to show that by affinity purifying the antibodies, you can decrease the IC50 of the antibodies against these heterologous parasites. And these antibodies also have much stronger blocking activity. They block the binding of AMA1 to its receptor RON2. And currently, what we are hoping to do is to be able to conduct this phase 1 clinical trial. And we've tried the matrix M adjuvant. You can see the same sort of somewhat subdued, but you can still see the same pattern of increased neutralizing antibody when you vaccinate with the complex in the matrix M adjuvant compared to the AMA1 alone. And hopefully next year, in collaboration with Patrick Duffy's group at LMIV, we hope to be able to conduct this the phase 1 clinical trial. So in the last few minutes, I want to just switch gears and present something quite unexpected and interesting that we saw. And this has to do with the ability of these AMA1 binding antibodies that block its interaction with its receptor RON2. And this was from our non-human primate study, where these monkeys that were protected from malaria challenge had a much stronger ability to block. This is an IC50 study looking at the levels of antibodies that block AMA1 interaction with RON2. So the lower the IC50 means greater the potency of the antibodies in blocking this receptor ligand interaction. Yeah? And so we found that there was a strong correlation between the presence of these AMA1 RON2 blocking antibodies and protection in this non-human primate study. And so we said, are such antibodies produced or induced upon natural infection? And working with our collaborator, Pete Crompton, and he has this beautiful longitudinal cohort where they collected samples before and after the malaria season. And we used about 1,200 of his cohort samples, 600 pre-season, 600 post-malaria season, and looked for this AMA1 RON2 blocking activity in these serum samples. Interestingly, we see sort of a bimodal response of these individuals in their ability of their serum to block AMA1 and RON2 interaction. And we took this cutoff point between these two bimodal points, about 64% of AMA1 RON2 blocking activity, as a cutoff to see to define as low or high AMA1 RON2 blocking activity. We see that with, like many other parasite antigens, the proportion or the levels of these AMA1 RON2 blocking activities increase with repeated exposure, which is not very surprising, right? And in data that I'm not showing, this would also correlate well with overall AMA1 antibody titers, which is again not that surprising. Interestingly, when you parse out the blocking activity as low versus high, individuals with high blocking activity have fewer clinical episodes, and they seem to be better protected against from infection with malaria, as determined by a parasitemia load of 2,500 parasites per microliter of blood. Okay? Here is the twist. It appears that the blocking activity is higher in individuals who are asymptomatic at the time of sample collection, right? So these are people who are blood smear negative, but you'll see that, and they had PCR positivity data, and you see that the individuals who are asymptomatic have higher levels of these AMA1 RON2 blocking activity compared to individuals who are negative at the start of the malaria season. When you account for all those variables, it's interesting to note that when you have high blocking activity, the presence or absence of falciparum parasites in those individuals seem to correlate with an increased, correlate well with protection. And it's the same thing the other way around. If you are asymptomatic, the presence or absence of high AMA1 RON2 blocking activity correlates with better protection in these cohorts, right? Okay. And the other interesting aspect was that when you have high AMA1 RON2 blocking activity, it seems to reduce the risk of fever. So this is the y-axis here is showing the percent individuals free of fever. And when you have high AMA1 RON2 blocking activity, the individuals have less chances of developing clinical symptoms, right? And here is another interesting part. It does not seem to affect the time to first parasitemia by qPCR, but once they are parasitemic, it delays or reduces the risk of developing the first malaria episode, the febrile episode. So I think this is the first, in my mind, the first evidence of a functionally defined type of antibody that correlates with reducing parasite burden in these individuals. And the last piece of data is when you compare, so this is an odds ratio. Now it's showing the odds ratio that when individuals start completely negative, PCR negative, and they end the season as PCR positive, compared to individuals who are negative throughout the transmission season, the individuals who become positive during the malaria season, the odds of developing AMA1 RON2 blocking activity increases, right? So this is, I think, close to a proof in my mind, suggesting that the presence of parasites over a period of time increases or helps in the development of these AMA1 RON2 blocking activities that together with potentially other parasite targeting antibodies work together in concert to reduce parasite burden. So why is that important? Why am I so excited about that? And one last data here is that if you can purify these AMA1 RON2 blocking activities from these two individuals, the individual who has high AMA1 RON2 blocking activity also seems to neutralize parasites greater than the individuals with lower AMA1 RON2 blocking activity. Again, showing that these naturally produced AMA1 RON2 blocking activities can neutralize parasites. Okay, why am I so excited about that? Simon talked about the durability of vaccine responses. AMA1 is one of the most immunogenic antigens, and this is the study from Chris Plow's work. Even though the vaccine trial did not work, what is important here is that individuals who are vaccinated with AMA1 and monitored over 360 days, the levels of antibody titers stays pretty flat. So from a vaccine development perspective, if you're able to induce the right type of vaccine responses and maybe even recall the right type of responses from these previously exposed individuals, maybe you can develop longer durable vaccine responses, is my hope. And my really last slide is we don't think that a single antigen is going to do it, right? So we need an antigen cocktail, and so we are exploring few other antigens that we can combine with our AMA1 RON2 or RH5-based mRNA vaccines to build a cocktail. So that's what we are working towards. Just to thank people in the lab, all the work that I showed you with the mRNA and the fusion protein work was done by the talented Sean Yannick, MD PhD student, and Varsha Venkatesh, who's just graduating from our master's and is joining Vaxxus company that delivers these silkworm-based needle-free injection techniques. Sam, who did the PF AMA1 work. Vani and Yue Lee, former people in the lab. Vani is actually in the University of Washington in the immunology department as a graduate student, and they did all the AMA1 RON2 blocking assays. Our collaborator, Marty Boulanger, our crystallographer in-house, essentially. Chuan Tran for all his help with the cohort study, as well as Pete Crompton for that work. Obviously, a lot of this work was initiated in Lou's lab, and I was a postdoc, and we continue to collaborate with Lou and Carol and her team as well. And thanks to Patrick and David at LMIV, who are spearheading the phase 1 clinical trials, collaborations with Drew Weissman's group at UPenn on the mRNA vaccine work. And I didn't have a chance to present the work from in collaboration with Ali Salanti that sort of piggybacks on what Simon talked about, the VLP platform of using the spy catcher, spy tag approach. And that certainly seems to increase the antibody responses, at least a log. So we are excited about that. So I'll stop there. have time for questions, but we can move on.
Thank you very much. Are there any quick questions? Michael? Do we have a?
Thanks very much, Prakash. Lovely. In the animal models where you vaccinated and seen protection, and when you challenge those vaccinated animals, whether they be monkeys or mice, do you get boosting of the antibody response?
Yeah, we have not tested it, but Simon did it for us, I should say. So from his RH5 trials, when they did, they also had an AMA1 arm, and they tested post-infection after parasites were cleared. They could see boosting in a number of animals for the AMA1 arm.
Good. Very.
Simon can correct me if I'm wrong.
Other questions? There's a question.
I have to ask this, sorry. mRNA versus the recombinant protein that you did. I mean, you said you didn't do dosing study. How do you know that, first of all, your recombinant protein is in the same intact form as the mRNA produced one? And could this just be an effect of better production from the mRNA to begin with?
Well, so...
And what's the protein level of the mRNA produced AMA1?
Yeah. So the only way we've qualified the mRNA-produced protein is through our Hexel expression study. So we've characterized that protein for its reactivity to specific conformational monoclonal antibodies. And they seem to recognize equally well the E. coli or Pichia-produced AMA1, the recombinant protein, versus the mRNA, the vaccine that generates proteins from the human cells. Oh, those sites have been mutated. We can talk later.
Okay.
All right, thank you.
Thank you very much.